Monica Gotta, and I lead a lab working on cell division and development at the medical faculty. My lab is interested by a fascinating question and a fundamental question. How can a complex multicellular organism originate from one single cell, the fertilized oocyte? We know this one cell will need to divide many times to create identical cells and more cells for our body. But these cells will also need to divide in an asymmetric manner to create cells that are different from each other and that will contribute to originate our organs and tissues. In the next few minutes, some of the people in my lab will take you through our projects that focus on cell division and development and cell differentiation, and will tell you that we address this question using two systems, the C. elegans embryo and human cells in culture.
Hello, I'm Ida and I'm a cell biologist. I'm interested in understanding how cell polarity is established. But what is polarity and why do we need it? Cell polarity is the intrinsic asymmetric organization of a cell. An establishment of polarity is crucial for the function of the cells and therefore for the function of tissues and our entire body. It has a fundamental role in many organisms, including humans, to create cell diversity during development and in adult life. In my work, I'm focused on the one-cell C. elegans embryos, where the part proteins, the main actors involved in polarity, become asymmetrically localized. And they form two different cortical domains, the anterior domain and the posterior domain. The goal of my project is to understand how polarity establishment is regulated, which means how this protein becomes localized on one side or the other of the one-cell embryo.
Hi, my name is Sofia. I am a physicist and a postdoc. The cortical polarity Ida has just described is translated into cytoplasmic protein gradients. One main question we have in the lab is about the physical and biological mechanisms behind the establishment of these gradients in the C. elegans one-cell embryo. Among the variety of different proteins that can establish a gradient, our focus is on a kinase which is of crucial importance for a successful cell division, the polo-like kinase PLK1. During asymmetric cell division, PLK1 exerts its function by being recruited at very different mitotic structures. Moreover, it becomes more and more enriched at the anterior cytoplasm. This leads to an asymmetric redistribution of this protein in the two daughter cells, the IB anterior cell and the posterior P1 cell. The aim of my project is to shed light on the complex dynamics of this protein by using an interdisciplinary approach combining mathematical modeling and advanced microscopy.
Hello, I'm Victoria and I'm a chemical biologist. As you have heard, PLK1 is an important kinase which regulates cell division processes like chromosome segregation, and it drives cell differentiation during development. One important feature of this kinase is that it localizes to different structures in the cell and during development. However, which pool of PLK1 is important for which function remains largely unknown. During my PhD, I'm making use of my skills as a chemical biologist to better understand the functions of PLK1 at the different locations in the cell and during development. For this, I'm combining organic chemistry and biology to make new tools that I want to use both in C. elegans embryos and in human cells.
Hi, I'm Simona and I'm a cell biologist. As you have heard, PLK1 plays many different functions in cells. To understand how PLK1 regulates these processes, we performed proteomics and yeast-to-hybrid screen to find regulators and substrates of this kinase. This led to the identification of PQN59 as a novel interactor of PLK1. Through my research work, we found that PQN59 plays a role in cell division, and it is also involved in the formation of stress granules in C. elegans, similarly to the human equivalent protein named UBAP12. We studied the fascinating process of stress granule formation in both human cells and C. elegans, trying to understand which are the proteins involved in the formation of stress granules, their structure, their role, and if PLK1 is involved in this process.
hope that you have enjoyed what you have learned from the people in my lab. As you have seen, we combine different approaches that include imaging techniques, biochemistry, forward and reverse genetics, mathematical modeling, and chemical biology. And we combine all these approaches with both model system, human cells, and C. elegans. We are looking forward to meet you and maybe to welcome you in our team. Bye.